S. Raja, Associate Professor, Department of Genetic Engineering, SRMIST of Science and Technology. I welcome everyone to the Membrane Protein Interaction Lab that is located in the 4th floor of the Bioengineering Block at SRM, Katangalotto. This lab has been established in 2017 with the support from the SRM management, and in 2018 we have received a grant from DST-SCRB around 40 lakhs. With that money we have further established the lab. In this lab, it primarily focuses on the membrane protein interactions, particularly and membrane biology. So we are interested in how the protein molecules are organized themselves, are specifically recruited to the membrane surfaces. So basically, I try to understand the language in which the protein and lipid talk to each other, how specific set of proteins are recruited, how this molecular level interactions do happens between the amino acids that are present in the protein and the lipid molecules that are present in the membranes. So in this regard, we are focusing on 3 different areas. One is the topology of the proteins that are present in the membranes, that we are studying using the guanine cyclase C as a model, and how we develop different model membranes, and how this protein insert, and how the amino acid composition as well as the lipid composition present in the membrane is going to influence the topology. The second key area of our lab is going to be the exosomes, the recent attractions for several cellular and molecular biologists. As you know that exosomes carry a multitude of cargo and perform a various roles like communication, sorting, trafficking, as well as regulating the cells. So we are going to studying the exosomes actually to understand disease pathology, development and progression. For example, one of my scholars, Ms. Archana, she is working on the exosomes derived from the DMD patients in urine and the blood samples, and she tried to understand the composition of the cargo and how these exosomes actually mimic the cellular physiology, so that we'll identify some markers to understand the progression of the disease. And another way, we also engineer the exosomes. So here we specifically change the proteins that are decorating the surface of the exosome, so that they could be specifically targeted to a particular cell, or we can also change the composition of the cargo by fusing the required protein to the markers already present in the exosomes. There is another scholar, Ms. Varsha. She is working in this area. She specifically engineered the tau protein, so that we can understand the tau proteins in a better way. So when you engineer the tau proteins with the exosomal markers, they'll get sorted themselves into the exosomes, and we can use them to study the tauopathies. And the other area of our work is we are also concentrating on the molecular ecology a little bit. There is one more scholar. He is working on the plant transcriptomics, where he try to understand the gene regulation networks and the other control elements that are present in the sorghum plant. So he is using the huge data that is already available in the internet using bioinformatic tools. D-Tech and M-Tech students also working in our lab. They are also working with the exosomes. Particularly, they are trying to understand the differentiation of PC12 cell into neurites, and they use the exosomes again to understand the differentiation process. They are fusing the CD9 protein with the fluorescence markers such as mCherry and GFP, and they try to use them to understand how this specific proteins are getting sorted themselves into the exosomes. And we have published papers and attracted funds, and I request people to visit our lab if you happen to be in the SRM University. Thank you very much.
I'm third year PhD scholar working under Dr. SRMIST in membrane protein interaction lab. My research focuses on identification of potential protein biomarkers from extracellular vesicles of Duchenne muscular dystrophy patients.
Hi, myself Nikhil Shinde. I am working on comparative transcriptomics and gene co-expression studies in sorghum. In present investigation, we are identifying key genes associated with the stem sugar accumulation in sorghum. This investigation will leads to this investigation will give interactive gene co-expression network that will facilitate better understanding of complex traits such as stem sugar accumulation. Thank you.
Hello, I am Harsha Singh, first year PhD scholar. My work mainly focuses on engineering exosomes, where we are studying trafficking of exosomes in neuronal cell lines.
Hi, I am Maheshwari, an MTech student working under Dr. N.S. Raja, and I am working on exosome engineering. And my work focuses on expression study of CD9 protein when transfected into HEK293 cells.
Final year BTech students, and we are doing a major project under Dr. N.S. Raja in MPI lab. We chose this lab because the works going on in this lab seem to correlate with our field of interest.
The project that we are working majorly focuses on the isolation of exosomes from differentiated and undifferentiated PC12 cells.
We are going to characterize the isolated exosomes of PC12 cells by the following methods of FTIR, HRTM and Western blotting.